So welcome. The whole point of this is to try and highlight, not only we're going to give a quick overview of current treatments for Dravet syndrome, many of you will be aware of those, but also highlight the new genetic trials that are on coming on board, and to give an opportunity for you to find out more about them and sort of timelines with regard to that, and maybe just how patients can get into those trials. So with no further ado, I'm going to share slides. I'm going to start off with the overview about where we are with current treatments for Dravet syndrome, and then I'll hand over to Andreas, who will talk about the natural history study that's coming on board, but also with regard to the clinical trials. Okay, and at the end, we think there will be lots of time, we hope, for questions. And you can ask them live, because this is not a huge webinar, or you can put questions in the chat and I'll moderate those to try and answer them. But feel free to do as you wish. So Dravet syndrome, I don't have to illustrate to this group, I'm sure, is an early onset developmental and epileptic encephalopathy. Children presenting in the first year of life, usually with prolonged seizures, possibly requiring ITU admission, and then the later appearance of other seizure types and developmental problems becoming progressively more apparent, with more than 80% being the result of a mutation in the SCN1A gene. In general, prognosis is unfavourable for seizures, cognitive development, and with a high mortality. And I mean, just to emphasise, I'm wondering whether we've only got paediatric professionals, but also the fact that this is not just a paediatric disease. They live into adulthood, and seizures are an ongoing problem, not least in this survey suggesting that only 6% of the whole age spectrum had been seizure-free for the past 3 months. And just to give the illustration of the developmental trajectories seen typically in this group of patients. Over the years, it's become evident about what treatments have been most successful, and valproate, clobazam with stiripentol, and possibly topiramate have been those that have been our staple, being aware that there can be seizure aggravation with certain medications, not least the sodium channel blockers. And of course, it was this population that we actually gained the first randomized controlled trial in a small, well-defined population with stiripentol, this now reported as far back as 2000, demonstrating the addition of stiripentol to valproate and clobazam being significantly more beneficial in reducing seizures than placebo. The longer-term safety of stiripentol has also been demonstrated with no evidence of waning of effect. There's been some discussion about whether there are particular populations that may respond better to stiripentol, and attempts to see whether the genetic mutation may influence it. Possibly there's greater efficacy when there is the mutation, help if I could spell. And also, as we now move into increasing recognition of adults with the syndrome, certainly efficacy has been demonstrated, although tolerability may be more problematic. And of course, then there's a range of open label studies with other medications suggesting benefit in some, not least with topiramate, particularly with prolonged seizures, levetiracetam, refinamide, and even more recently, perampanel. But also older medications, and there are certain centres that use bromide, can be really helpful where other medications have indeed failed. And we mustn't forget the ketogenic diet, which in this study demonstrated an efficacy almost as good as standard treatment with clobazam, valproate, and stiripentol with regard to responders. Yes, we've become aware of treatments that we need to avoid, not least the sodium channel blockers. This wasn't only highlighted by Renzo Guarini's team, but others have also demonstrated, not least Andreas in their studies. But we've also learned that it may affect natural history in this study, demonstrating better cognitive outcomes in those where contraindicated medication was not used in the first 5 years of life, compared to the population where carbamazepine had been used. But it's never too late, and that has only been too rightly illustrated by Sanjay Sisodia's evaluation of diagnosed with serious adults with Dravet syndrome and the SCN1A mutation in a population in Chalfont. And interestingly, they found 22 newly diagnosed adult cases, the oldest of which was 60 years. 7 had drug changes following diagnosis, and of the 3 who had meaningful follow-up, 2 had improvement in cognition and 1 spontaneous language for the first time, and this was actually the 60-year-old. But this said, it's also caution, because there do appear to be some individuals who are responsive to sodium channel blockers. This a report of 3 adults, or teenagers and adults, where there was a seizure exacerbation on withdrawal of lamotrigine. So we do need to show caution. And of course, not to forget the practical aspects with regard to prompt treatment of fever, the avoidance of hypothermia, and the awareness of triggers, not least excitement and possibly photosensitivity, particularly in the very young, and having individualized protocols for the prompt treatment of prolonged seizures, although I'm not going to discuss emergency management here. What about the newer agents? Well, of course, cannabidiol has been illustrated both in the original study in 2017 and this more recently published study last year. The first study, an evaluation of a single dose of cannabidiol versus placebo in a population with Dravet syndrome, and in the second study, 2 different doses of cannabidiol versus placebo, both achieving the primary endpoint with a significant % reduction in seizure difference between cannabidiol treated and indeed placebo treated. But what was interesting is that the cannabidiol higher dose of 20 milligrams per kilogram per day did not achieve much greater benefit than the 10 milligrams per kilogram per day. And there were more in the way, as I'll come to, of adverse events in the higher cannabidiol group. Adverse events were not particularly problematic. There were similar percentages with having all cause in both the cannabidiol and placebo group, although somnolence, diarrhea, decreased appetite and fatigue were the most common side effects in the cannabidiol group. And now we have the open label extension studies, which also demonstrate that there doesn't appear to be a waning of effect. There is continued benefit over time. There is, of course, the ongoing issue about interaction with clobazam, originally pointed out in the open label studies. Increased norclobazam levels being noted, and that may end up with some side effects. But the original dose finding study demonstrated that actually in the presence of stiripentol, there is no such increase in norclobazam levels, demonstrating a saturation effect. And of course, now we have fenfluramine studies, whether the individuals are on stiripentol or not on stiripentol, demonstrating significant benefit in seizure reduction in randomized controlled trials over placebo, and the difference between the active medication and indeed the placebo being very wide, unprecedented in what has been seen in previous studies. Side effects were not a particular concern, particularly with regard to decreased appetite, that there may be concern about, or weight decrease. There was, and fluramine, of course, there was the original concerns about cardiac effects when it was utilized in the 1960s, which is why it went out of favor. But certainly in the studies to date, no such problems have been noted, although ongoing cardiac surveillance will be required. There's also the issue about whether there are benefits with the fenfluramine beyond seizures. There was the brief undertaken in the studies, and yes, in subscores, benefits were seen in the fenfluramine group, which were not seen in the placebo group. But this was relatively short term, and we have to wait to see whether other benefits may be seen. But one other interesting point is we know there's a high rate of mortality, particularly in the young, in this particular group of patients. This showing the, in this cohort study by Cooper and colleagues, a SUDEP incidence of 9.32 deaths per 1,000 patient years in the Dravet group. And if you look at the trials prior to the use of fenfluramine, was a similar SUDEP rate, which dramatically reduced in the fenfluramine active group post utilization of fenfluramine in the trials. Whether this is due to the reduction in convulsive seizures or another mechanism of action is yet to be determined. So here we are with the possible algorithm we might consider. First line being valproate, with then the addition of stiripentol with or without clobazam. And currently, depending on the approvals, with cannabidiol, thereafter with clobazam. But now fenfluramine has also been licensed within England. There is a process going through with regard to NICE, the HTA approval, and that's yet to conclude. But it would be envisaged that hopefully it will be certainly up as second line, and whether it lies above cannabidiol, of course, will come online dependent on our experience with use. So with that, I'll hand over to Andreas to talk about where we are with the natural history study proposed, and ultimately the genetic clinical trials that are coming online.
Thank you, Helen. That's very kind. So we're exactly quarter past 6, which is half time. So really, so it's really my great pleasure to talk to you about, first of all, about the SCN1A Horizons study, so a natural history study of SCN1A related epilepsies in the United Kingdom. And I think it's really important that we start really assessing our patients in great detail. Do you want to go to the next slide? Is that okay? Yeah, so really, I think what's just to remind us all about the pathogenicity of Dravet syndrome, and that this really is a developmental and epileptic encephalopathy. And of course, we have the epilepsy, but we have lots of comorbidities. We've got a movement disorder with ataxia, but also we've got learning difficulties and autistic features. So there's really much more. Dravet syndrome is much more than just an epilepsy, really an developmental epileptic encephalopathy. And it's really important that we actually measure all of this to have a better idea whether treatments truly are effective. Next slide, please. So because Dravet syndrome is one of the most common childhood genetic epilepsies, it's really a model disease to study this. And we now, as Helen so eloquently portrayed, we've got emergence of drugs effective at reducing seizure burden. However, we don't really know what the impact on cognition and on development is. And really, without the understanding of the natural history study of Dravet syndrome, I think it will not be possible to really measure treatment success just beyond seizure control. And we really need to characterize the disease phenotype in detail. Next slide, please. So the SCN1 Horizons natural history study will be able to allow us to provide standardized and better care for our patients tailored to their needs, because the aim here really is that all children included across the United Kingdom will be assessed in similar ways, which we will hope will also raise the standards of care. And so this is a joint working with the neurologists and neuropsychologists in 27 different centers, a collaborative effort across the UK. And with really an aim to integrate the assessments into standard clinical practice to really minimize the extra burden for the clinical teams. So next slide, please. And so in terms of recruitment, where we are with SCN1A Horizons, so we're aiming to start in the autumn of this year, and the study will run over a period of 4 years. And any child or adult with Dravet syndrome or SCN1A related epilepsy can participate. Patients can be new patients or established diagnoses. There's a total of 300 participants of all age groups, stratified into 240 children and 60 adults. We thought it was really important also to gather natural history data on adults as well. So it will be across United Kingdom, England, Scotland, Wales, and Northern Ireland. And the participants are allowed to be enrolled in other studies or trials at the same time, which is also really important. Next slide, please. And so what does this study actually entail? So there will be 2 different types of visits. First of all, the clinical visits as part of the routine care. This will be every 6 months. And 1 month prior to the clinical visit, the patients and parents will fill in a seizure diary prior to the appointment. And then during the assessment, during the clinical visit, there will be a structured clinical record form that will be completed. And then there will be an additional visit, which is a neuropsychology visit. And this will be either every 6 months or every year, depending on the age of the child. So for the younger children under the age of 6, 6 monthly assessments. For the ones 7 years and older, yearly. And these will be detailed face-to-face assessments regarding learning and behavior, and also questionnaires regarding comorbidities and quality of life, really to capture the whole phenotype of SCN1A-related epilepsies. Next slide, please. And so what we really hope that we can achieve with this SCN1A Horizons natural history study to get a better understanding of the epilepsy phenotype and the neurodevelopmental outcome over time, to identify biomarkers, because we don't have many biomarkers, and predictors of disease outcome, and by including patients into this natural history study to raise the standards of care for individuals with Dravet syndrome or SCN1A-related epilepsies. And we would think that being part of a natural history study network that streamlines also access to best evidence treatment approaches. So that leads us to the next slide, really focusing on new disease-modifying gene-based treatments in Dravet syndrome and SCN1-related epilepsy. So next slide, please. So just a reminder in terms of setting the basics of the sodium channel alpha subunit, so this is a transmembrane protein, and we know that proteins are the building blocks of the body. We've got over 400,000 proteins in the body, and the proteins consist of amino acids, and we know that the SCN1A protein is a very long protein and contains 2009 amino acids. And really the master plan how to assemble each protein is contained in our DNA. The next slide, please. And so really we understand the DNA as really the recipe book for the proteins of the body. Next slide, please. And what you can see here, so each DNA recipe really is called a gene. Here you have, and this is really quite important as well later to explain how the different disease modifying treatments actually work. So we've got treatments that work at the level of the gene, then the gene is the DNA is transcribed into the mRNA. So we've got treatments that are working on mRNA and then translated into proteins. And so it's just important to understand here also how the different disease-modifying treatments work. So next slide, please. Sorry, yeah, and so really, and there's now a whole range of different approaches for disease-modifying treatments. And so if we start at the basics, where we know that an individual has a pathogenic SCN1A variant or mutation, which is then might result in, for example, a truncation of the protein, so the protein is not functional anymore. We know that this leads to a developmental disorder with epilepsy. And then we've had a number of rodent models and also human models to really explore disease modifying therapies, which includes a range of either RNA-based therapies, you might have heard about CRISPR, or AAV9 vector-mediated treatments. And we go through these in detail now. So next slide, please. And this is a really nice overview. This grabs from an overview of Stephanie Shorgus and Gabriele Lagnini's paper in neurochemistry in 2020. So I would encourage reading that, because it gives a really nice overview. And here just gives you in the round diagram, so the several principles of how you can have disease-modifying therapies either via CRISPR or via AAV9 vectors and gene supplementation, or by addressing the RNA and RNase therapies. And here I will just start with approaches that use, for example, CRISPR gene editing. So CRISPR really is based on bacterial CRISPR-Cas9 defense system, and so the cell's genome can be cut and at the desired location, allowing existing genes to be removed and or new ones to be added. And one technique that Gabriele Lignani and his team are really using now is CRISPR-A, so it's gene activation using CRISPR, and then uses a Cas9 protein, which is adapted with a transcriptational activator to modulate endogenous gene expression. So this is really a technique to increase the endogenous gene transcription and thereby to increase the levels of SCN1A protein that's produced. If you move to the next slide, please. That's really illustrated here in terms of the CRISPR rationale, for example, for Dravet syndrome. So here at the top, you can see that in physiological and a normal person, you will have two SCM1A gene copies, and they both produce 50%, leading to 100% gene product. However, if you have in Dravet syndrome, one of these is faulty, and so only 50% of gene product and protein product is actually available. And what can now be used in the gene activation by CRISPR-A is really to increase the endogenous gene transcription and to increase the amount of protein that is actually produced, and thereby reaching levels of 100% of SCN1A protein. So this is evidence from animal models. However, there are no studies in humans yet. So moving to the next slide. So the next approach is called targeted augmentation of nuclear gene output, or TANGO, which has recently been published in Science Translational Medicine, and it's been led by Stoke Therapeutics. So it's an RNA-based treatment using antisense oligonucleotides, also called ASOs. And here it's delivery via repeated intrathecal injections, so repeated lumbar punctures. And the aim here really is to restore the protein levels by increasing the protein production from the functional copy of the gene. And I explain in the next few slides how this actually works. And this really leads to selectively boosting the expression only in tissues where this protein is normally expressed. And this technique can also be used to address small or large gene targets. So if we move to the next slide, this is courtesy of Stoke, who provided these slides. So really illustrating here the haploinsufficiency without the TANGO ASO treatment. So what you have here in green, you've got normal wild type, and in red, you've got a mutated gene with loss of function. So on the left-hand side, in green, you have your normal wild type that then gets transcribed into the pre-mRNA, and there's a process of splicing, which then leads to the productive mRNA production, and that leads via translation to the protein. And what happens is that we have these NMD, nonsense-mediated decay exons, which lead to non-productive mRNA, and then RNA degradation by nonsense-mediated decay. And then you have the mutated gene, which doesn't produce any protein output. So this individual here with Dravet syndrome has only 50% of the functional protein output. Moving to the next slide. So what the TANGO ASO therapy actually does, it promotes the NMD exon exclusion. So this nonsense-mediated decay exon gets excluded, and thereby you augment the production of productive mRNA, and thereby have the increased production of functional protein, which means that you can correct the deficit that's been presented due to the mutated gene that doesn't produce any protein output, and therefore you correct this. And if you move to the next slide, you can see here then in mouse models. So this is STK001 is product, and that really significantly reduces the premature mortality in mice, and that's after a single dose. And what you can see here in the graph is the survival percentage of mice, and you can have in green you've got the STK wild type, and yellow you've got the treated STK001, and in red the untreated mice. You can see that the survival of the untreated mice in red clearly is shortened, and what you see with time, what you can see here is that with these with the treatment, there's clearly prolongation of mortality, of survival in these mice. Then moving to the next slide. So what's the current status of the STK001 trials? So these are, Stoke has conducted the first in-human dosing in Dravet syndrome patients, which started last year in the United States. And currently it's the Monarch trial. So it's a phase 1/2 trial that's currently ongoing in the US and aiming to recruit 48 children and adolescents between the ages of 2 and 18 years of age with Dravet syndrome and confirmed SCN1A variant. Here in the UK, we're planning now for the, or Stock is planning for the Admiral study. That's again a phase 1/2 trial planned in the UK to recruit about 22 children, adolescents, ages 2 up to 18 years old with Dravet syndrome and confirmed SCN1A variant. And the endpoints, so these are phase 1 and 2 trials. So the endpoints are mainly safety and tolerability of ascending doses to make sure that these are actually safe to administer. And they will measure changes in seizure frequency, overall clinical status, and quality of life. And the aim is to start recruiting later this year. Moving on to the next slide. So the another treatment approach has been the GABAergic cell-specific SCN1A gene regulation, and that's been driven by encoded therapeutics. And so it's a DNA-based treatment that is an adenovirus AAV9 delivered. And this is a one-time intracerebroventricular, so ICV administration into the ventricles. So this will require a new surgical procedure to administer the product. And this contains a regulatory element targeting specific GABAergic cells and then an engineered transcription factor to upregulate SCN1A expression within the target cell types, which are GABAergic inhibitory interneurons. Moving on to the next slide, please. So this ETX-101, so it's a cell-selective gene regulation therapy approach addressing the underlying cause of SCN1A positive Dravet syndrome. So what you can see here, so you can see the AAV9 vector that then contains the regulatory element targeting GABAergic cells, and then the transcription factor to augment, to increase expression. And so it's really a gene regulation therapy, and it's adeno-associated virus mediated. And this will then, due to the vector, transported into the cell, into the nucleus, and then leading to increased production of mRNA and proteins. And if you move on to the next slide. So the, and this is just an illustration of the schematic representation of the engineered transcription factor that really will bind to the DNA and has then the effect of recognizing a unique sequence upstream the SCN1A transcription start site, and then thereby increasing the production of mRNA and more SCN1A protein. If you move to the next slide. So in animal models, this also has shown that this treatment is successful in preventing hypothermia-induced seizures and normalizes the survival in Dravet mouse models. Here on the left-hand side, you can see, so the survival, you can see in dark blue, these are the uncontrolled Dravet mice, and in lighter blue, the SCN1A treated mice with ETX-101, so the survival is better. And again, in terms of hypothermic seizures, here what can be shown is that in the SCN1A controls, there's more hypothermic seizures, whereas the treated mice behave almost or close to the wild type. So moving on to the next slide. So where we are currently with the encoded STX-101 trial. So that trial is going to be called the Endeavour trial. Again, it's a phase 1/2 trial, which is planned internationally, including the US, UK, Australia, and Spain, and involves 24 children aged between 6 months and 4 years with Dravet syndrome and confirmed SCN1A variant. Again, so this is a phase 1/2 trial. So really the endpoints here are primarily safety and tolerability of ascending doses to make sure this is safe. And then also measures of change in seizure frequency, the mental outcomes, behavior, motor function, sleep, and quality of life. And so the aim for recruitment would be that this starts early in 2022. Next slide, please. So really, we've seen a lot of development in terms of treatment for Dravet syndrome. So we've got an increasing number of evidence-based treatments available for Dravet syndrome. And I think what Helen also illustrated earlier, that the treatment algorithms are really likely to change over coming years. And we have the new disease-modifying gene-based treatments becoming available. So we've got the Admiral trial starting in the UK this year, later this year, then the Endeavour trial starting in the UK next year. And then we have the SCN1A Horizons UK Natural History study also starting later this year, and really hoping to improve standards of care with children with SCN1A related epilepsies and Dravet syndrome. And this leads me to the end of the presentation. And just to highlight, there's the conference in November, chaired by Helen, and would be a very informative venue to obtain further information with regards to new therapies and where we are with treatment with Dravet syndrome. Thank you.